Did you know that some of the deadliest substances on Earth have become our most powerful medicines? Imagine a toxin so precisely engineered by evolution that it can shut down your nervous system in minutes. Yet that same molecule, slightly modified, now saves millions of lives each year. Today, I'll reveal the 15 most remarkable venoms that transformed from weapons of death into instruments of healing. Poisons so sophisticated that pharmaceutical companies spend billions trying to replicate what nature designed over millions of years. From a snail that inspired the most powerful, non-addictive painkiller ever discovered, to a lizard whose venom became a breakthrough diabetes treatment, each represents a paradox: beauty and lethality, danger and salvation, nature's cruelty repurposed as mercy. Stay to the end, because number 1 is a venom so medically revolutionary that it has spawned an entire class of life-saving drugs used by hundreds of millions of people worldwide, all derived from a creature most people would kill on sight. Let's begin. Number 15. Gila monster venom, exenatide. Picture the Arizona desert at twilight, and crawling across the sand comes a living mosaic of black and orange beads. The Gila monster, one of only 2 venomous lizards in North America. For centuries, indigenous peoples warned against this creature's excruciating bite. Yet hidden in that venom was exendin 4, a peptide that would revolutionize diabetes treatment as the drug exenatide. In the 1990s, endocrinologist John Eng discovered that Gila monster venom contained a hormone remarkably similar to human GLP-1, which stimulates insulin production. But here's the breakthrough: human GLP-1 breaks down within minutes, making it useless as a drug. The Gila monster version lasts for hours because the lizard's metabolism is incredibly slow. The venom needed stability in a reptile that might eat once a month. This evolutionary adaptation translated perfectly into human medicine. Exenatide helps diabetic patients control blood sugar without dangerous hypoglycemic crashes. Since its 2005 approval, millions of patients use it, with some versions requiring only weekly injections rather than daily insulin. The Gila monster nearly went extinct before we discovered this medical treasure. If we'd driven them to extinction, this breakthrough would never have been found. That's Gila monster venom, a desert reptile's slow metabolism teaching us how to manage blood sugar. Proof that medical breakthroughs hide in unexpected places. Number 14. Tarantula venom, pain research. Imagine venom so precisely targeted, it can teach us which specific nerve channels control pain. Tarantula venom contains hundreds of peptides, each targeting different receptors, and researchers are using these toxins as a molecular toolkit to map the human pain system with unprecedented precision. The Peruvian green velvet tarantula produces ProTX2 that specifically blocks NaV 1.7 sodium channels crucial for pain sensation. Humans born without functional NaV 1.7 feel no physical pain at all. Those with overactive channels suffer burning pain so severe some contemplate amputation of uninjured limbs. The tarantula toxin acts as a precise molecular probe, allowing researchers to study these channels and develop drugs that might block pain without affecting other nerve functions. What makes tarantula venoms valuable is their specificity. Each peptide evolved to target particular prey species. This evolutionary arms race created a natural library targeting virtually every nerve channel type. Modern pain management relies heavily on addictive opioids that work broadly on the nervous system. A medication derived from tarantula venom could potentially block pain at its source without affecting brain centers, creating the first truly novel pain medication in decades. That's tarantula venom. Hundreds of peptides teaching us how pain works. Spiders as unwitting research partners in fighting chronic suffering. Number 13. Scorpion venom. Cancer treatment. Imagine venom that glows under ultraviolet light and can cross the blood-brain barrier, one of medicine's most frustrating obstacles. Scorpion venom from the deathstalker contains chlorotoxin that specifically binds to cancer cells, especially gliomas, brain tumors notoriously difficult to treat because they infiltrate healthy tissue with microscopic tentacles. Chlorotoxin's natural function is paralyzing prey by binding to chloride channels in nerve cells. But researchers discovered this same peptide has unusual affinity for cancer cells, binding to them far more readily than healthy cells. Even more valuable, it crosses the blood-brain barrier, a membrane that protects the brain but blocks most drugs. Medical researchers have synthesized tumor paint, chlorotoxin linked to fluorescent molecules causing cancer cells to glow during surgery. Surgeons can visualize exactly where tumor cells are, seeing cancer invisible to the naked eye, allowing more complete removal while sparing healthy tissue. Beyond visualization, researchers are developing chlorotoxin as a delivery vehicle, attaching anti-cancer drugs to the peptide so they're carried specifically to tumor cells, like guided missiles seeking cancer throughout the body. That's scorpion venom, a deathstalker's toxin lighting up brain tumors, ancient predators teaching us how to see and destroy cancer. Number 12. Platypus venom. Antimicrobial peptides. Picture one of nature's most improbable creatures, the platypus, a mammal that lays eggs, has a duck's bill, and venomous spurs on its hind legs. Male platypuses produce venom during mating season, delivering a cocktail causing excruciating pain that conventional painkillers don't touch, leaving victims incapacitated for days. Yet hidden within are antimicrobial peptides that could help address antibiotic resistance. The platypus split from other mammals over 160 million years ago, and its venom contains defensin-like peptides with unique three-dimensional structures, potentially effective against bacteria that developed resistance to conventional antibiotics. These peptides work by disrupting bacterial cell membranes, a mechanism bacteria find much harder to develop resistance against compared to antibiotics targeting specific enzymes. Research reveals platypus venom peptides show remarkable stability and antimicrobial activity against both gram-positive and gram-negative bacteria. Their evolutionary history, splitting from other mammals before modern immune systems evolved, created peptides with similar functions but different structures, providing researchers with alternative design templates for new antimicrobials. That's platypus venom, an evolutionary oddity producing peptides that could solve antibiotic resistance, nature's most improbable creature offering answers to modern medicine's most pressing challenge. Number 11. Jellyfish venom. Cardiovascular research. Imagine trailing tentacles up to 120 feet long, each armed with millions of microscopic harpoons that fire faster than a bullet, injecting venom into prey. The box jellyfish is considered one of the most venomous creatures on Earth, capable of killing humans in minutes through cardiac arrest. Yet components of its venom are now being used to understand heart function and develop cardiovascular treatments. Box jellyfish venom contains pore-forming toxins creating holes in cell membranes, particularly targeting heart and nerve cells. The venom causes potassium to leak from cells while allowing calcium to flood in, disrupting electrical signals that coordinate heartbeat. This mechanism, while lethal in envenomation, teaches researchers crucial information about cardiac ion channels. Medical researchers use purified venom components as research tools, applying controlled amounts to isolated heart cells to study exactly how cardiac rhythm is generated and maintained. These toxins act like molecular scalpels, allowing scientists to selectively disrupt specific ion channels and observe results, creating detailed maps of cardiac electrical function. This research has contributed to developing better treatments for arrhythmias affecting millions. That's jellyfish venom, the world's fastest biological weapon, teaching us how hearts work. Creatures that kill in minutes, providing insights that save lives. Number 10. Funnel web spider venom, stroke treatment. Picture Australia's Sydney funnel web spider, often called the world's deadliest arachnid. So aggressive it rears up when threatened, fangs dripping with venom capable of killing humans in 15 minutes. The venom attacks the nervous system, causing massive neurotransmitter release and respiratory failure. Yet this same molecule is showing remarkable promise as a stroke treatment. The breakthrough came when researchers discovered the Hi1, a peptide blocks acid-sensing ion channels in brain cells. During stroke, when blood flow is interrupted, cells become oxygen-starved and die, releasing acid that activates these channels in surrounding tissue. This creates a cascade of destruction. Acid signals trigger calcium influx, causing more cell death, releasing more acid, expanding damage far beyond the initial affected area. Hi1A acts like a circuit breaker, blocking these channels and preventing the cascade. In experimental models, administration up to 8 hours after stroke onset, far longer than current treatments allow, significantly reduced brain damage. This is revolutionary. Current clot-busting drugs must be given within 4.5 hours. Hi1A works through a different mechanism and appears effective even hours later, potentially extending the treatment window. Clinical trials are now underway testing synthetic versions in human stroke patients. That's funnel web spider venom, a neurotoxin that kills in minutes being repurposed to save brain tissue. Evolution's weapon becoming medicine's shield against stroke. Number 9. Leech saliva, anticoagulants. Watch a medicinal leech attached to skin, its 3 jaws creating a Y-shaped incision, feeding for 20 to 30 minutes, while the wound bleeds for hours afterward, thanks to a cocktail of over 100 bioactive compounds in leech saliva. For thousands of years, leeches were used based on flawed theories about balancing bodily humors. But modern research has validated their medical value for entirely different reasons. The star compound is hirudin, an anticoagulant so potent it's now produced synthetically and used in patients who can't tolerate heparin. Hirudin specifically inhibits thrombin, the enzyme converting fibrinogen to fibrin during blood clot formation. Unlike heparin, which requires other blood proteins to work, hirudin acts directly, making it more predictable and effective for procedures like heart surgery and dialysis. Modern microsurgery has brought leeches back into hospitals for salvaging reattached body parts. When surgeons reattach fingers or ears, they can reconnect arteries but struggle with tiny veins. Blood flows into reattached tissue without adequate outflow, causing congestion and tissue death. Medicinal leeches remove excess blood and inject anticoagulant saliva, keeping blood flowing while new vessels regrow. Reattached body parts treated with leech therapy have significantly higher survival rates, up to 70 to 80% success compared to 30 to 40% without leeches. That's leech saliva, a blood feeder's toolkit, becoming modern medicine's anticoagulant pharmacy. Creatures once used through ignorance, now employed through understanding. Number 8. Cone snail venom, ziconotide. Beneath tropical waters hunts a creature dressed in geometric elegance, the cone snail, its shell a masterpiece of mathematical patterns. The geography cone snail fires a venomous harpoon that instantly paralyzes fish, delivering hundreds of peptides called conotoxins. One became ziconotide, a painkiller 1,000 times more powerful than morphine, non-addictive, and effective for patients whose pain nothing else can touch. Ziconotide works by blocking N-type calcium channels in the spinal cord, preventing pain signals from reaching the brain without affecting opioid receptors that cause addiction, tolerance, and respiratory depression. It's administered directly into spinal fluid for severe chronic pain when all other treatments fail: cancer pain, neuropathic pain, conditions where even high-dose opioids provide no relief. The cone snail evolved this toxin to freeze prey instantly. We've repurposed it to freeze pain signals. Each cone snail species produces unique venom cocktails, over 700 species, each potentially containing 100 to 200 unique peptides. Researchers have studied only a fraction, yet already found compounds for pain management, epilepsy, heart conditions, and mental health disorders. That's cone snail venom, a tropical hunter's paralysis toxin becoming medicine's most powerful, non-addictive painkiller, beauty and lethality in a spiral shell. Number 7. King cobra venom, neuroprotection. Picture the world's longest venomous snake, 18 feet of muscle and menace, delivering enough venom in one bite to kill an elephant or 20 humans. King cobra venom contains neurotoxins that shut down breathing by blocking signals between nerves and muscles. Yet researchers discovered certain peptides show remarkable neuroprotective properties, potentially useful for treating Alzheimer's, Parkinson's, and other neurodegenerative diseases. The peptide ohanin binds to nicotinic acetylcholine receptors, the same receptors dysfunctional in Alzheimer's. In laboratory studies, ohanin-related compounds protected neurons from toxic effects of beta-amyloid plaques, the protein tangles characterizing Alzheimer's. Other king cobra venom components show potential for protecting neurons from excitotoxicity, damage caused when neurons are overstimulated by excessive neurotransmitter release, a process involved in stroke, traumatic brain injury, and various neurodegenerative conditions. King cobras need their venom to work on prey nervous systems with surgical precision, so their toxins evolved to interact with neural pathways in highly specific ways. This specificity makes them valuable research tools and potential therapeutic templates. That's king cobra venom, a breath-stopping neurotoxin teaching us how to protect the brain, the world's longest venomous snake, contributing to longevity of human neurons. Number 6. Saw-scaled viper venom, eptifibatide. In deserts from Africa to India lives a snake responsible for more human deaths than almost any other. The saw-scaled viper, small but aggressive, its venom causing severe hemorrhaging as blood loses its ability to clot. The venom contains disintegrins preventing platelet aggregation. causing uncontrolled bleeding. Yet from this hemorrhagic venom came eptifibatide, a drug that prevents heart attacks by doing exactly what the venom does, stopping platelets from clumping. Heart attacks occur when platelets aggregate at sites of arterial plaque rupture, forming clots that block blood flow to heart muscle. Eptifibatide, derived from saw-scaled viper disintegrins, binds to platelet receptors and prevents them from sticking together during cardiac procedures like angioplasty and stenting. It's administered to thousands of patients daily during emergency cardiac interventions, significantly reducing heart attack damage and death rates. The irony is perfect. Venom that kills by causing bleeding now saves lives by preventing deadly clots. The same molecular mechanism, applied in different contexts. In a bite wound, it's catastrophic. In coronary arteries, it's life-saving. That's saw-scaled viper venom, a hemorrhage-causing toxin repurposed to prevent heart attacks. Evolution's blood thinner becoming emergency medicine's clot preventer. Number 5. Stonefish venom, pain pathway research. Imagine stepping on what appears to be a rock, only to feel the most excruciating pain humans can experience. Survivors describe stonefish stings as pain so overwhelming they begged for amputation. Pain that morphine barely dulls, that can last for months. The stonefish, perfectly camouflaged on the ocean floor, has dorsal spines that inject venom when stepped on. And that venom is revealing secrets about how severe pain operates at the molecular level. Stonefish venom causes immediate tissue necrosis and activates pain receptors through multiple pathways simultaneously. It damages cells directly, triggers inflammatory responses, and activates specific pain channels. Researchers studying why stonefish stings are uniquely agonizing discovered novel pain pathways and mechanisms we didn't know existed. The venom contains a unique combination of proteins that work synergistically. Individually, each component causes moderate pain. Together, they create synergistic agony. Understanding this synergy is helping researchers comprehend why some chronic pain conditions are so resistant to single-target treatments and why combination therapies might be more effective. That's stonefish venom, the ocean's most painful sting, mapping the neuroscience of agony. The worst pain teaching us how to relieve all pain. Number 4. Brazilian wandering spider venom, erectile dysfunction. In Brazilian rainforests hunts the world's most venomous arachnid, the Brazilian wandering spider, whose bite causes priapism among its terrifying symptoms of neurotoxicity and potential death. Yet researchers investigating why this occurs discovered peptides that could revolutionize treatment for erectile dysfunction. The peptide TX2-6 increases nitric oxide production in erectile tissue, causing blood vessel dilation through a mechanism different from Viagra. Significantly, TX2-6 appears to work even when conventional ED medications fail, and preliminary studies suggest fewer side effects. The peptide is also being investigated for treating cardiovascular conditions since it affects blood vessel function throughout the body. The medical potential emerged from asking why this particular symptom occurred, turning an embarrassing side effect of envenomation into a therapeutic lead. Clinical trials are ongoing, with synthetic versions being developed that maintain beneficial effects while eliminating toxicity. That's Brazilian wandering spider venom, a deadly neurotoxin accidentally revealing new approaches to vascular medicine, nature's most awkward symptom becoming a therapeutic opportunity. Number 3. Sea anemone venom, autoimmune disease. Anchored to coral reefs, their tentacles swaying like underwater flowers, sea anemones deliver neurotoxins that paralyze small fish instantly. Yet hidden within these ancient venoms are peptides, revolutionizing our understanding of autoimmune diseases like multiple sclerosis, rheumatoid arthritis, and lupus. Sea anemone toxins called Escherichia peptides selectively block Kv1.3 potassium channels overactive in certain immune cells responsible for autoimmune attacks. In autoimmune diseases, the immune system mistakenly attacks the body's own tissues. Kv1.3 channels in effector memory, T cells drive these attacks. Escherichia peptides can selectively suppress these harmful immune cells while leaving protective immunity intact. Dalazatide, a modified version of Escherichia, is in clinical trials for multiple sclerosis, psoriasis, and other autoimmune conditions. Unlike current immunosuppressants that shut down the entire immune system, dalazatide targets only the specific immune cells causing autoimmune damage, maintaining the body's ability to fight infections. Sea anemones, creatures barely changed in 500 million years, carry within their tentacles molecular solutions to diseases that have plagued humanity for millennia. That's sea anemone venom, ancient cnidarians teaching modern immunology, creatures older than dinosaurs showing us how to calm overactive immunity. Number 2. Assassin bug venom, Chagas disease research. Picture an insect that earned its name through methodical hunting. The assassin bug, stalking prey with patience, then striking with a curved proboscis, injecting venom that liquefies internal tissues. But some species carry something far more dangerous. They're vectors for Trypanosoma cruzi, the parasite causing Chagas disease, affecting 6 to 7 million people primarily in Latin America. The bug's venom has become a crucial tool in understanding the very disease these insects spread. Assassin bug venom contains enzymes that break down cell membranes and proteins. Medical researchers discovered these enzymes could break down protective barriers that T. cruzi parasites create, making parasites vulnerable to immune system attack or drug treatment. Additionally, researchers are studying how certain assassin bug species evolved resistance to the parasites they carry, hosting millions in their digestive tracts without becoming sick. The venom also contains anticoagulants and anesthetics. The bug needs to feed on blood without being detected. These components are being studied for cardiovascular disease and localized pain management. The same insect that spreads a devastating disease also carries within its venom potential tools for treating that disease. Biology's dark humor, creating parasites, and their potential cures within the same organism. That's assassin bug venom, a predator's digestive weapon repurposed to fight the disease its relatives spread. Number 1. Pit viper venom, ACE inhibitors. Picture a Brazilian pit viper called the jararaca, coiled in rainforest shadows, its venom containing a peptide that drops prey blood pressure catastrophically, causing circulatory shock and death. In the 1960s, Brazilian scientist Sergio Ferreira isolated this peptide and discovered it worked by blocking angiotensin-converting enzyme, AC, the enzyme that raises blood pressure. This single discovery launched ACE inhibitors, a class of drugs that has saved more lives than perhaps any other medication in history. The first ACE inhibitor, captopril, was approved in 1981, directly modeled on the jararaca venom peptide structure. ACE inhibitors revolutionized treatment of hypertension, heart failure, and kidney disease. Before ACE inhibitors, heart failure was a death sentence. Now, it's a manageable chronic condition. Millions take ACE inhibitors daily. Drugs like lisinopril, enalapril, ramipril, often without knowing they're derived from snake venom. The impact is almost incomprehensible. ACE inhibitors have prevented hundreds of millions of heart attacks, strokes, and cases of kidney failure. They've extended countless lives by years or decades. Healthcare economists estimate ACE inhibitors have saved trillions of dollars in medical costs and prevented suffering beyond calculation. All from studying why pit viper venom causes blood pressure to plummet. The discovery almost didn't happen. Ferreira was studying bradykinin when he noticed the venom's blood pressure effects. He pursued this observation despite skepticism from colleagues who thought studying snake venom was primitive and irrelevant to modern medicine. His curiosity about a side effect of venom from a common Brazilian snake led to one of the most important drug classes ever developed. If Ferreira hadn't been curious, if the jararaca had gone extinct before we studied it, millions who are alive today would have died from cardiovascular disease. This isn't hypothetical, it's statistical certainty. The jararaca pit viper, unremarkable except to herpetologists, carries in its venom glands the molecular blueprint for controlling blood pressure in hundreds of millions of humans. That's pit viper venom, from rainforest killer to humanity's guardian. A common Brazilian snake that has saved more human lives than any doctor, any hospital, any medical advance you can name. From pit viper venom that became the most prescribed cardiovascular drugs in history, to cone snails providing painkillers more powerful than morphine without addiction, to Gila monsters teaching us how to manage diabetes. These 15 venoms share a profound truth. Nature has already solved many problems medicine struggles with, and evolution is the most sophisticated drug designer we'll ever encounter. What strikes me most is the pattern. Every breakthrough came from curiosity-driven research into organisms most people would kill on sight. Scientists studying why does this venom work this way, accidentally discovered treatments for heart disease, diabetes, cancer, stroke, chronic pain, and autoimmune diseases. None came from trying to develop drugs. They came from trying to understand biology. The conservation implications are staggering. We've studied perhaps 0.1% of venomous species, analyzed a tiny fraction of venom components, and already found treatments used by hundreds of millions. How many other medical breakthroughs are we losing every time a species goes extinct? Every time a habitat is destroyed? These venoms reveal a humbling truth. We're not as different from other animals as we'd like to think. The same ion channels, receptors, and enzymes that venoms target in prey exist in humans. When a cone snail paralyzes a fish, it's targeting the same calcium channels we have. When a pit viper drops prey blood pressure, it's affecting the same ACE enzyme regulating our cardiovascular systems. Perhaps most importantly, these discoveries teach us that we can't predict where the next breakthrough will come from. Who would have guessed that studying spider bite side effects would lead to ED treatments? That jellyfish capable of killing in minutes would teach us how hearts work? That the world's deadliest spider would provide stroke treatments? The question these venoms leave us with is urgent. What are we losing right now? What species are going extinct before we can study them? What venoms contain the cure for Alzheimer's, the solution to antibiotic resistance, The key to treating cancer. We can't know what we're losing until it's gone. And then, it's too late. These 15 venoms are both warnings and invitations. Warnings that biodiversity loss isn't just an environmental issue, but a medical crisis. That extinction forecloses therapeutic options we haven't even imagined. And invitations to approach even the most dangerous, frightening creatures with curiosity and respect. Because within their venom glands might lie the molecular keys to saving millions of human lives.